Okay, so it's called triplet because one sequence of 3 bases codes for one amino acid. So we talked about DNA replication. Now we're going to talk about transcription and then translation. Hopefully we'll have enough time to do the lab. Okay, so the DNA is in the nucleus and it cannot leave the nucleus. But the ribosomes are out in the cytoplasm where the proteins get made with the ribosomes. So then the DNA has the recipe to make the proteins. It has to get that recipe out of the nucleus and into the cytoplasm. And the way it does that is by making an RNA copy of the DNA, which is called the messenger RNA. And that can leave the nucleus and go out into the cytoplasm, where the ribosome can make the proteins using that recipe. So if we look at a DNA strand, I'm just making this up as I go along. Okay, so there's a double-stranded piece of DNA. For any given protein, only one of these strands contains information, and we call that the template. We'll say it's this one in this case. Say it's the template strand. This other strand is just there. It protects the bases and it helps the DNA replicate itself. Okay, we saw how that happens. So only one of these is going to be containing information. So what's going to happen, again, an enzyme is going to separate the base pairs. And I'll just get rid of this strand for now. They're going to be separated. And another enzyme is going to bring in complementary RNA nucleotides. So let's find the red pen. Because red seems to be RNA's color. Okay, so this is DNA. So in RNA language, what's going to be complementary to A? U to G, C to A,
U
to C,
G
everybody to A,
U
to G,
G
keep going, U,
U A U U C C.
So on the RNA, this is now what's called a messenger RNA. And it gets decoded by the ribosome in triplets. So 3 of these RNA sequences are called codons. Okay, so, and we have codon tables that we're going to use to decode them.